Thank you for joining us today, this afternoon, and to the actually worldwide audience. I'll welcome online now as well. So to officially kick off the Alzheimer's Insights AI Prize, the finalists for the $1 million competition, and as we just heard, a second prize as well. Thank you, Stephen. So just to set the stage for today, some points here. We have 5 really incredible teams presenting that some of you here got a preview already. I myself have been impressed over the course of this 7-month journey, not only the passion that they brought, but the speed which they've accelerated their innovations from the initial round to a semifinal round to today. It's been really incredible to see how these teams have engaged in the platform. To set some expectations, each team will have 15 minutes to present, and that includes a live demo. And afterwards, myself and my fellow judges, both here and online, will be asking them 5 minutes of questions each. All teams will be attempting a live demo of their AI agent. And as anyone in tech knows, that takes a lot of courage and bravery to do. And so I admire all the teams for attempting to pull that off today in a time-bound presentation. We do have a video backup if any of those go south, and that's okay too. But really applaud the teams for trying to do that for us here today. So the teams, after their 15 minutes of presentation and demo, will be taking live questions, as I mentioned. It'll be from the judges only. However, you will. We really want input not only for the teams and feedback for them, so you can scan the QR code during their presentation here and provide feedback, which will go to the teams, and they'll provide answers as they go forward. So during each of the QA session, you'll see a QR code that you can scan. Please provide your input or questions there. Those will be provided to the teams, and we'll give them that feedback. We want to make sure you share that feedback with the teams for the summit. So with that, I don't want to take too much more time. I do want to just thank my judges here today, both in present and online. This judging panel represents expertise from AI to biotech to hyperscalers from the tech industry to venture. It's an amazing group of judges with experience that are going to lean in and really make a really tough decision today. So with that, I'm just going to go in order here. Thank you to my judges here. So Elena Alfiglione from Microsoft, Zoe Cortzi from Cambridge, Justine Levin Ellerand from Flagship Pioneering. Sorry, Justine. And then online we have Ashima Gupta from Google, and then Tom Stocky from Facebook and Google as well, and is also a senior advisor for GRIP. So thank you to all. Can I have some applause for my judges who turned out here today? So with that, without further ado, I do want to kick off this competition and welcome our first team to the podium. So Team Fingerprint, the stage is yours.
It's really a pleasure to be the first ones here to actually start this pitch session. We are Adrian and Arvid, and we actually believe that Alzheimer's disease is the defining biomedical challenge of our generation. We are researchers at MIT and the Broad Institute, and founders of Y Combinator-backed companies. To address this challenge, we're building Fingerprint. Fingerprint is a discovery acceleration engine composed of specialized agents and new foundation models that interpret biological signals to enable prevention and treatment by helping us find novel biomarkers, prevention interventions, and therapeutics. A fingerprint. What does a fingerprint do? It uniquely identifies a person. All of us have it. That's why we use it in biometric collection, solving crimes. In biology, the same idea applies. Each of us carries a biological fingerprint, basically a unique combination of signals that reveal disease risk and if properly understood, enable prevention and treatment of Alzheimer's disease. The story begins a little while back, and let's take a step backwards and look how this field has evolved. In 2015, the finger trial showed that multi-domain lifestyle interventions can slow cognitive decline. That discovery launched the Worldwide Fingers Network. Now it's 72 trials, well, actually now 73 trials across 40 countries and over 30,000 participants, one of the larger prevention ecosystems in medicine. The past February, last year actually, we launched Fingerprint, which was built to actually go and build a global discovery engine. And this past February, we partnered with the Davos Alzheimer's Collaborative to take that impact global to different regions in the world, and that includes India, Latin America, Africa, among others. And what is so astounding for us is how fast this has evolved. In last October, when the idea of the pitch competition started, we were just in an inception mode. It was just very conceptual. And despite very limited resources during that period of time, we have evolved, our team has evolved extremely fast. This has moved at a very, very fast pace. So we started building discovery engines and new foundation models that can reason against those global data sets. And that incredible progress has actually now includes a multi-omic foundation models, 11 papers, AD workbench integration, and partnerships to support that global access. Fingerprint now lives in the AD workbench, and the next phase focuses on taking Fingerprint as a translation, bridging those models into real-world cohorts and clinical prevention trials. But again, we have to step backwards a little bit and think about the Alzheimer's disease field and how fragmented it has been in terms of pushing discovery forward. Alzheimer's disease, as I mentioned, is deeply fragmented, so we have genomics data, proteomics data, clinical cohorts, and intervention trials, which each exists still in pretty much isolation. So instead of answering one big biological question, we end up answering very small ones. And that's a problem because Alzheimer's is not a single pathway disease, it's a system biology problem. Other fields have solved this challenge. We heard during the previous panel some examples of those successes. And for example, we can think about the oncology field, where by integrating all of these different modalities and data, that revealed that breast cancer was not a single disease. It was actually many diseases, and each had its own biology and its own treatment. And that shift transformed the outcomes. If you go a century back ago, breast cancer survival rates were very, very, very low. Today, more than 90% of patients survive early-stage disease. And the same story is beginning to happen in metabolic diseases. Like if you remember the trajectory that we see in metabolic diseases, we can actually know and understand that biology starts to precede this decades before the onset of those symptoms. And the same goes for Alzheimer's disease. We know very well, we're in this conference where we know very well that disease begins decades before cognitive symptoms emerge, which means that the real opportunity lies in early detection, primary prevention, secondary prevention, combination therapies, guided by precision biology. But to go there, we first need to solve this fragmented discovery.
With Fingerprint, our goal really is to deliver the same step change that we've previously seen in oncology to Alzheimer's. Where we are as a field right now is we've collected lots of data. We have prevention trials. They generate genomics, proteomics, metabolomics, clinical signals. But all of this is just too complicated. We basically have no real novel fundamental insight. We've built Fingerprint to make sense of all this. We orchestrate a set of specialized agents and completely new foundation models we've built to really integrate this multimodal data with clinical trial data to fundamentally accelerate how we discover new diagnostics, preventive interventions, and new therapeutics. The tools we've developed include Finger7b, the very first multi-omic foundation model that really accelerates the discovery of new diagnostics and prognostic biomarkers. This is also one of our papers, recently accepted at iClear. You'll get to see a preview here in the QR code. An agent that really automates the discovery of new therapeutics. And our thinking microscope, which is an entirely novel tool that really enables fast and accurate search for multi-omic biomarkers. That's work currently under review, but you'll get a preview here in our data room as well. And all of the source code for these tools is publicly available for reproducibility and works across platforms, including the AD Workbench, so all of you can use it. Let me walk you through Finger7b, which is our 7 billion parameter multi-omic foundation model. Recently published this at iClear, and the source code is also publicly available, so really all of you, independent of the outcome of this prize, can use it. We pre-trained this foundation model on 8 trillion genomic and metabolomic tokens and genomic tokens that we've assembled from large public data sets, and we've introduced a whole new method that we call quality-aware tokenization, also one of our recent papers, to really turn those extremely noisy and heterogeneous multi-omic data sets to something foundation models can actually learn from. And that's how we've assembled the very first multi-omic pre-training corpus. Just in a few weeks, really, we fine-tuned this model directly on data from clinical trials like the Worldwide Fingers, ADNI, and BioHermes. And now we've applied it, and what we can share with you are some extensive benchmarking results. We've compared against major frontier models like Metagene and Evo2. What we see really is that Finger7b outperforms these models on all general benchmarks like next token prediction, can handle multimodal prediction and classification tasks like both metagenomic profiling and metabolic pathway prediction, something that really shows the fundamental limitation of some of the existing models, which are trained on a single omics modality and can't handle multimodal prediction tasks at all. For example, Evo2 completely lacks the metabolic representation. Now, we further validated this on real clinical trial data. We really see our incredible performance improvements, like around 100x better few shot efficiency, 13x improvement in stratifying responders, 20% increased accuracy in AD detection. And that's all really based on retrospective analysis of real clinical trial data, so that's incredibly exciting. Now, let me show you some of the work we've done just in the past few months, really highlighting the accelerated discovery by Fingerprint. We've already worked with Worldwide Fingers, and just over the past few weeks, we've really set up an additional MOU with Doug to amplify what we can do globally. And based on all this, we pioneered a whole new idea called a clinical trial in the loop. So whenever our model generates a new hypothesis, we pull in data from participants of some of those large trial networks to validate it, and new trial data directly feeds into our model. And that's how we essentially enable continuous learning and real-time discovery across those global cohorts. Ultimately, our goal is to enable model-guided trial design and use novel discoveries enabled by Fingerprint to really build better trials. So currently, ongoing trials always teach the model improves future trials. That's a whole new reinforcement loop that's really never existed before. And in the spirit of truly multimodal analysis, let me walk you through an exciting example of what we've done on the therapeutic side. We'll dive in, we're looking to understand, are there targets that we can identify from these data sets? As a case study, we identified TREM2, and using all of our in silico tools, we're really able to take that target and develop early in silico predictions of potential molecules that have great blood-brain barrier penetration that could target this. This is early, it's in silico, but we want to take this forward and validate those new molecules. And what this really gives you is a sense of how, using fingerprint, we're able to massively accelerate the discovery of those new therapeutics. And this incredible pipeline from really multimodal data ingestion to, for example, here, mechanistic target and therapeutic candidates, all AI-driven and really autonomous. Now let's see a quick demo where we'll have a look at this in action. This lives fully inside the AD workbench. And we'll have a look here at the full pipeline, really made up of 6 distinct agents that ingest super complex, raw, multimodal data and turn it into mechanistic insight that directly helps us discover new biomarkers and therapeutics. One of the exciting agents we have here, for example, is our thinking microscope. Whenever we're looking for genomic, metagenomic biomarkers, we often don't need to look at the entire data set, but we can apply clever sparsification methods to really reduce the amount of data we're looking at to massively accelerate the identification of those new biomarkers while retaining accuracy. We also have our structural reasoning agent, which is the TREM2 example we've previously seen. And now let's run the whole thing. We'll start with a few data sets that we've prepared, for example, 100 worldwide fingers. We prepared a few examples here with the details available in all our related papers. Once we've input all these raw, really multi-omic data sets, we get to run the entire pipeline and see individual contributions. For example, here we've built our own knowledge base, really tapping into all the relevant work that's been done in the space. Our thinking microscope to really look for genomic, metagenomic specific biomarkers. We've identified new molecular targets. We have our own really metagenomic multi-omic foundation model here that gives us detailed longitudinal predictions and personalized treatment summaries. And we've come up with this really detailed personalized recommendation here where we recommend specific interventions and predict treatment response. Now, while this fully works in the AD workbench, we've also built our demo, which is publicly available here, and you should try it out as well. Let's look into a super novel contribution that we have here as well, our own foundation model called Finger 7B. Let's take a closer look what we can do here. We can, for example, go ahead and score a multimodal sample. We've come up with our own risk quantification methodology, really based on reading in this full patient sample here, around 50 million reads, with detailed predicted longitudinal prediction of cognitive decline and a detailed prediction of up to 11 years. We see in detail, for example, here the specific metabolomic and microbiome changes present in this patient, and including, for example, here with this additional multi-omic foundation model, the microbiome is super important, especially if we're doing multi-domain interventions. We can, for example, go ahead and do let's see what we can recommend for intervention. If we look at both metformin and lifestyle, we get to see, for example, that patient's microbiome composition before and after the intervention and predicted response. And that additional modality, for example, here at the metabolic profile and the microbiome is incredibly important if we're, for example, including multi-domain interventions in this combination therapy that include dietary changes as well.
Perfect. And that's fingerprint. Basically, an engine composed of special agents that Arvid just showed and foundation models that interpret those biological signals and accelerate the discovery of novel biomarkers and therapeutics. And what we see here is basically the community that makes it all possible. You see here partners across academia, biotech, and industry in this really collaborative spirit. Some of them are actually here, and we have Miyaki Pelto, who directs the Worldwide Fingers Network, Li Huaisei from the Pike-Hour Institute, we have Anna Matum from Karolinska, Gio Traverso from MIT, and our colleagues from Novo Nordisk and Alamar that are also present. In that foundational spirit, really, this is what we think that a global collaboration really needs to accelerate discovery, both in therapeutics, being non-pharmacological and pharmacological. And at the end, what we really need is combination of both of us, both of those. So as we know, Fingerprint is already running in the AD Workbench ecosystem, and we're working hard to expand that entire discovery pipeline to really make it global, and that's where our collaboration with the Davos Saldo Collaborative comes to play. Basically, this is a tool that we hope that it goes for the community, not only for our team, and that's why we have a demo.fingerprint.bio for all of you, and also the integration with the AD Workbench. And now we're really focused on the commercialization aspect that is of this really, really already for us, very impactful research. So thank you to all, and hope that you can join us as well later this Friday, where we're going to do a demo live in one of the booths. Thank you.
I'll open this up to questions.
So you speak to the importance, of course, of identifying mechanistic insights in order to drive better discovery and better therapeutics. And the multi-omic data that you have and other sources that you brought together obviously drives those mechanistic insights. One of the features that you have on the platform is druggability. So the druggability of the target. What additional data do you think we need to bring together in order to reinforce that druggability score, so that way we actually can create the therapies needed to target key pathways?
Absolutely. So as we showed at the beginning, during that beautiful scheme, or the different types of modalities that we're including, for sure already we have genomic and proteomic data, but we believe that for the druggability piece, we need to bring on top of that, we're talking about the gut microbiome, we're including pieces of that, but we would like to include as well pieces of treatment response from others that have already included. And without the network, there's examples of that. We showed you metformin and lifestyle. We can actually determine the response to treatment, and that would be a very valuable piece of information that we can add as well. Mm-hmm. There are certainly other modalities. We're actually doing something not related to that. So I just mentioned genomic and proteomics and others, but there's something we're including other modalities. We're working with a team in India to include speech as part of this multi-omic, really multimodal aspect.
Hi. Thank you. Really interesting. So continuing the multimodal kind of theme, can you tell us a little bit about how you maybe integrate co-pathologies, and you can ask questions about counterfactuals in this kind of pathway all the way from discovery to clinical trials? So
we include from baseline, we include clinical history. That's already your first point of entry. Based on that clinical history, that patient at baseline, you have an idea of their previous, at least self-reported, and then you have as well the medications that they're taking at baseline. Usually those are individuals that are not in a non-dementia stage. So they are very early because we're targeting that kind of primary prevention, but also a bit of secondary. So those are included in the model for taking account for the accreditation. Now, those co-pathologies might behave a bit different depending on the region where that patient originates. And that's where we want to bring all of that global aspect with other regions where we don't know exactly the behavior of that multimodal aspect with or multi-omic aspect with the clinical trajectories, which might change, in India, in Kenya, in other regions, because at the moment everything has been very centered either in North America or Europe. So that's where we want to incorporate that. But at the moment we're using the kind of baseline clinical data that's already coming with the patient at entry.
All right. Yeah.
Just a follow-up. So can you ask questions, given that you're building it?
Sorry?
Okay, sorry. So given that you're building this for clinical trials and looking at response, right, drug response, can you build in counterfactual questions? So for example, what if a patient came from a different region or whatever, they had different pathologies, or, what if they have changed their lifestyle and now they're going on to a drug target?
Just a quick response, then we'll finish. Go ahead.
This is something we're already doing, and you can check it out in our demo as well. That's essentially the fine-tuning of our model to integrate these additional questions. So we're going to do this kind of with few-shot learning, where we see that our model performs really well to ask those counterfactual questions, and really, out of all the ways you could do this, we believe it works best if we build models that understand clinical trials on a molecular and genomic level that's really representative of entire populations, and that's the foundation model we've built here.
Thank you.
One last quick question here.
Yes, thank you. So congratulations, first of all. How interpretable is the model to neuroscientists or clinicians in terms of what it explains versus what it predicts, and when do they know which one?
Do try it out at our public website. I think that will be incredibly helpful to see that we provide detailed mechanistic insights and the most important features, really, that went into making the decision. So you get to see the full reasoning and chain of thought that lead up to the treatment recommendation. We've also built our own LLM essentially to ask follow-up questions and make that easier for clinicians to directly use.
So just very briefly, in that aspect, we are actually requesting additional feedback from all of you to bring that forward. We're actually using the price of the funding, 50% for deployment and further optimization of the AD workbench, but a lot of that is also going to cross-validation, like 30%, and the final 20% is really to support that our teams global.
Well, lots of interest here and lots of questions. Let's just thank the Team Fingerprint for a great presentation. And so we'll keep us moving along here, and I'm going to welcome Team Alethea to the stage. Welcome.
Hello, everybody. This is Yiyu, and I'm Travis, and we're representing Team Alethea. We're going to be talking about our agentic AI platform for Alzheimer's disease drug discovery. So the real issue here is that and the reason that we want to automate the ADRD drug target nomination and drug discovery process is that there's a couple major bottlenecks in this process. One is that it's incredibly time-consuming, and the second is that there's a very high technical barrier involved with doing this type of research. One of the reasons that it's actually time-consuming is that there might be, or there is, quite a bit of manual literature review that needs to take place to identify gaps and also form hypotheses. And also to cross-reference analyses across different databases and different omics types. So this is obviously very, very time-consuming, and this leads to only a couple targets being nominated per year. The other issue, which is also tied into this time consumption, is the fact that there's a very high technical barrier for people to conduct this type of research. So for you to basically be able to identify these AD drug targets, you need to have some proficiency in bioinformatics, machine learning, programming, and also some AD/RD subject matter expertise. So just to orchestrate these complex pipelines across very heterogeneous data and very heterogeneous patient populations, it does take a lot of training. So one thing that we can do to actually improve this is that we can actually develop this agentic AI mechanism. So one thing that we can do is that we can improve this drug discovery process, nominating more targets per year, partially because we can lower the technical barrier. So by using these modules that we've implemented using an agentic AI platform, we can actually reduce the amount of programming that's involved and use more point and click and user interface so that more users can actually use this worldwide. Another thing that this actually helps is people in low and middle income countries be able to do more AD research. All right.
Okay, so basically, one of the fundamental questions we were thinking about when we are developing our tool is, why is this agentic AI $1 million question for this particular ADRD drug discovery, right? I mean, agentic AI has been very, very hot topic. It has been applied to many, many general areas. So why is this particularly important and difficult to be applied in the ADRD drug discovery research? So we think that there are two fundamental barriers. Number one, you have to gain the trust from the ADRD drug researchers, right? And number two, they have to be able to use the whatever agentic AI tool efficiently. So for the first part, we think the most important fact is not how powerful this agentic AI tool is, but rather how grounded, how transparent, how trustworthy this model is. And number two, the model has to be easily deployed, even for the researchers who have zero knowledge in AI, machine learning coding, and they have to run efficiently without having to ask them to buy like $1 million server, waiting for a long, long time to get things set up. So based on that, we kind of designed our tool using three blocks. The first block being it's a knowledge base, and the second block being an LLM-based router. We use the GPT-4o as the router for the language model router. And then finally, a toolbox of 5 domain tools. So basically, our flow works like this. We have a user that would send a plain language, like text request. For example, can you do the study this AD versus the normal for the data set we're going to upload? And then the router will start to analyze the request. It's going to generate an ordered and concrete action plan. For example, I'm going to call the harmonize tool to do the data normalization, to do the QC, and then I'm going to using another tool to do the clustering, and then I'm going to call, for example, the hypothesis to generate the hypothesis that can be tested based on the data set provided. So this whole action plan is viewable by the user, and our agentic AI will also provide justification why this is being suggested. And then upon execution, instead of letting this language, like the router, to run everything in a black box, in a blind setting where the hallucination problem can occur, they can generate something that's out of nowhere. We iteratively query on the knowledge database that we created. This is another highlight of our tool. It's a 2-layer knowledge base. We call it evidence cached twin knowledge graph. One layer will be the paper layer. We would query or retrieve from the large body of literature online, from the Semantic Scholar, from PubMed, related to the particular study. We also can also retrieve from the database, like the Go, Campbell, for example, all these data sets. And then, so this iterative query is being done through every step, so that we're making sure that every step is grounded, that they are trustworthy, they're based on the evidence. And also, in order to make things efficient, because this query can take time, we inspired this from the computer science perspective, where we develop a cache kind of like architecture, where previous queries are being cached in the local. So if it's a hit, meaning that we have already done similar queries before, then it's going to directly return from the local cache. And if it's a miss, it's a new query that has not been done before, we would go online in real time to pull the relevant information. And then once we return the results, we're going to store them locally as well. So this even looks complicated, but it only adds a minimum overhead to the computation. And also, we have a total of 5 different tools listed here. They handle different things. For example, they do the clustering, the general hypothesis based on the data set. The KB prune is what we use to validate against the knowledge graph. And then the code gen is used to generate code. If we find that existing tools cannot cover what's being needed, our agentic AI can write its own code to do that. And so once the user sends the request, we handle everything. And upon return, the results being returned, can also be viewed by the user, whatever the results are, like the DE tables or whatever results. The user can say, hey, I'm satisfied, I'm done, right? Or I'm not satisfied. I think maybe we can tighten the QC bound, or we can just adjust whatever parameter. That can be provided back to the router in plain text. The router will adjust all the parameters automatically and rerun all the experiments. So our tool can either be run as a fully automated tool, or user can take control, become an interactive tool. Both modes are fine. Next slide. And then we take everything one step further, right? So in addition to just asking the user to specifically describe, hey, this is what I want to do, in a very specific task, why don't we just let the agentic AI do its own research? This is very hot today, right? And we believe this is the first fully automated research engine for ADR drug discovery, where the user would just have to specify, hey, I'm interested in, for example, microglia lipid panel, lipid metabolism. So based on that description and based on the data user uploaded, we have a total of 5 collaborative agents. They work together to do the research. So first of all, the first agent right here, where this ideation is an engine where it would go online. It would check all the relevant papers, like millions of papers in PubMed, in Semantic Scholar. It finds the relevant papers, it analyzes the paper, and identifies the research gap. What are the potential gaps? It's going to generate hypotheses that can be tested based on the data uploaded. And then it's going to design, the next agent will design the experiments, hey, what kind of experiment needs to be run, what kind of result needs to be collected. And then the next agent will actually start to try to use the existing tools, and if needed, it can generate its own code if the experiment cannot be run on existing tools. And that's also very important because that particular part, we know that LLM can generate code that might have bugs. The agent will identify by reading the traceback and see, hey, there's a bug, this is the problem. It will self-correct, self-heal the error, and then regenerate the code and rerun in a fully automated way until everything's fine. We also have after every step, we also ask the agent to ask itself, is this biologically reasonable, right? Again, we don't want to do something that's not reasonable. And if the agent identifies, hey, this is something that's not really reasonable, it's going to backtrace and take one step back, go to the previous step. So we make sure by doing this, we make sure every step is fully grounded in evidence, not based on hallucination. And eventually, the output will be, we generate a report. It can be in any text format. For example, if you want, hey, can you write a paper to NeurIPS? Just provide a template. It's going to give you a NeurIPS paper with all the charts, all the tables. It's going to also give you the code being generated so you can rerun everything for full reproducibility, for full traceability. So everything can be fully traced and audited. And also going to give you the figures, the rationale, so everything will be provided. And also you can just basically with one click, you can start from your simple description of idea to multiple hypotheses, to being, they can be handled in a batch with all the experiments, all the analysis, and finally to all the papers. That only takes one click. All right, so that's our idea.
And the reason that this is all a great fit is that we can actually make all of this kind of end-to-end. So instead of having these individual steps, we kind of tie it all together using our agent. So this takes fragmented pipelines to end-to-end pipelines, ungrounded evidence to evidence-backed at every step. And obviously, this is going to make things a lot faster through automation, which in turn also makes this very scalable. So some of the data sets that we're currently using are single nucleus RNA-seq data sets and multi-omics data sets, but there's really nothing stopping us from using a very, very diverse set of data across many different databases. And these can include, upon proper data use agreement, access, things like AMP-AD, but across a multitude of different databases, including even chemistry databases to improve druggability.
Okay, just give me one. I'm launching the extra demo, so just give me one second. Let me just make sure.
Yeah, so one of the reasons that we actually want to use so many data sets is obviously druggability is a huge issue. So if we can get a better sense of whether or not, for instance, something could, pass the blood-brain barrier, or if, there's a possibility that a small molecule will bind to a specific protein that we're targeting, all of these pieces of information can improve the probability that we can get a proper therapeutic.
Okay, I'm done. So can you switch to the demo? Yes. Okay, so as I just explained, our tool actually has 3 different modes. The first mode will be single omics. It can handle the specific task for single omics, then the multi-omics, and then finally the ASR, which is the fully automated research system. So let me start with the single omics. So this is the interface of single omics, and it has a field on the left showing the drag and drop the files here. This is where you would upload your data. So for example, let me go to here, the data demo. For single omics, our tool can basically handle any main type, like the 4 main types listed here: epigenomics, metabolomics, and proteomics, and single cell RNA sequence. I'm just go with one of them as for the demo purpose. You just drag and put the data here. It's going to be uploaded. And you can see here, there is a box saying that, hey, can you enter your analysis request? Oops. This is where you would actually upload, enter your information. And also to save time, I have also pre-entered the prompt here. So let me just do copy and paste. Let me just copy everything. Copy and paste here. So you can see that's just plain language. It doesn't require any programming. And the task here I'm asking for is, can you nominate top 20 possible drugs? And basically for analyzing AD versus normal. And also we have a setting here. This is where you can either just manually select the tools you prefer to use. We have a few tools you connected already, or we'd recommend just use the AI to decide the best tool, best flow. So in this case, we are just using the best tool available, and then that's everything, right? So then you click on send. This whole thing starts to run on the back end, like AD Workbench. So integrating AD Workbench. There's nothing you need to kind of program. You can just copy and paste our code and the package into your workspace, and you can just run the whole thing without writing a single piece of code. So it's analyzing everything, and this process takes just a little bit over a minute. It's pretty efficient, and you are not waiting it's going to print all the intermediate steps. For example, right, you can see here, it's giving you a very detailed plan. What am I going to do? It's giving you the justification. Why am I recommending you to do these? And it tells you, hey, every single step for the analysis, if you click on that, it's going to also allow you to visualize all the intermediate result. It's going to tell you all the the thresholds, all the parameters being selected. It's also going to tell you why. It's going to give you justification. So for example, right here, what are the decisions made by the agent, and why are these decisions made with a clinical relevance? So they are not being generated out of the blue. So by doing this, we're minimizing the possibility of hallucination, which is well known for most of the large language models. So you can see that we have a lot of different steps going. Now we have it's running the hypothesis. So just to save time, I will go to the result. This is the result we were able to generate just to save time. This is the final result I can show you for that analysis. You can see the list here, a total of 20 targets being nominated. And you can see that in addition to giving these results, we are also giving an evidence score. So the definition of evidence score. Yeah.
Really rich presentation, but I need to leave some time for questions.
Just maybe 30 seconds?
5 seconds. Go ahead.
Okay. So basically, you can just click on that, and then it's going to give you every information needed. Especially, we also provide the druggability and the relevance to all the literature and to all the database, so you know how trustworthy and how relevant these results are. And we can do the same thing for the other two. It's the same process, just you get all the results.
Thank you very much. Some questions here. I'll start out first. So can you just describe who you think your first users of this would be? What would they be?
Yeah, so I can actually take this one. We're in a couple of AD drug discovery initiatives at Indiana University. So we're actually proposing some of our targets from this pipeline to them, and they will, design some, knockout experiments and also potentially even look at small molecules to, see what we can do with some of these targets. So we're actually trying to prospectively validate the results from this pipeline, and we have a plan for that.
Thank you.
Thank you very much for your presentation. So shifting from faster breakthrough and productivity to real new insights, what did the model give you as specific new biological insights that would not have been available if one would have done traditional research processes?
So I think one aspect to this, in terms of improving the speed at which you can do analyses, is it actually improves the depth, right? You can do more analyses. You can look at more, comparisons, right? So I mean, we've already, identified certain AD biomarkers like GFAP already. But there's also things in that list that, to my knowledge, I've only seen in a couple publications like Lingo1 that actually, seems like it might be, a relatively good drug target. So I think, the efficiency does improve the number of things that you can identify. But we've already kind of identified a few things that we want to pursue further. So I think we are addressing that.
Thank you. Just a quick one. So you said the model knows what is biologically relevant. How does it know? What if it's a new discovery and we've never seen this combination of markers, for example? How does it know?
So I think that's an excellent question. I think some of it, actually takes that human in the loop, right, to, make educated decisions about this. That's why we've kind of built a human into the loop in this framework. One thing that I think that you have to take into account is if there is a lot of biological reference, like it has a very high score because of the literature, that's not going to necessarily be novel. So you might want to actually look at things that have a high score except for, the literature evidence. And all of this is taken into account, and you have all of that information in the final reports.
And also, I want to add this a little bit. So the capability of LLM, as evidenced by many of the literature, including, for example, the AlphaFold, people have demonstrated that language models are capable of kind of discovering or predicting things that are not in the literature yet. They can do that pretty well. So that's the kind of main driving force behind our engine. We hope that we can utilize the prediction capability. And in fact, for that single, the one that the demo I just showed, there are a couple of them that are not studied or well studied in the literature. And from our group study, we found that they are potential good targets. So we are already seeing that evidence in our preliminary results that I just showed in the demo.
Thank you so much. Really compelling presentation. I'll remind the audience to submit feedback with the QRS code, and let's thank this team right here.
Thank you.
Now, I have the privilege of welcoming Team Cortex to the stage. You have your 20 minutes, 15 minutes for presentations and 5 for questions.
Thank you, Greg. Hello, everyone here in person and online. Today, I will talk about a problem that affects millions of families and a new kind of AI that addresses it at scale. This is Alzheimer, an agentic AI ecosystem that, for ADID that transforms the interplay between clinical care and scientific research. ADID has 2 deeply interconnected challenges. First, most patients lack access to timely personalized care. For example, more than half of the patients never receive a timely diagnosis. Over 85% of patients are left without post-diagnosis care, and only about 9% are ever invited to a clinical trial. This is not a niche gap. The ADID care delivery challenge is real, large, and urgent. Our answer is solution part 1, patient care AI agents. a set of agents that support patients and their physicians across all stages of ADRD care, from risk prediction, early intervention, all the way through diagnosis, clinical trial management, and long-term care management. But there's a second challenge running in parallel. We have rich ADRD data, but the translation from data to care is far too slow. On average, it takes over 2 years just to get from data collection to a publication, and many more years from this scientific finding can really benefit a patient in a clinic. This traditional path is too long, and our data could help patients right now. That's why we introduced ADRD AI co-scientists. They help close the data to care gap and bring research data to patients and their physicians. What makes our product fundamentally different is the connections between these two layers. They do not operate in isolation. Real-time scientific discovery can flow directly from AI co-scientists to patient care AI agents. And real-world clinical insights can feed back to improve the clinical relevance of ADRD research. This bidirectional intelligence drives co-involving intelligence between patient-facing AI and researcher-facing AI, far beyond anything a standalone AI project can achieve. This is the first two-layer agentic AI ecosystem of this kind for ADRD or any complex disease. Here we go through each layer. On the patient-facing level, we have agents cover the decade-long window of ADRD care with the physician in the loop. For example, one patient may ask, how much can I reduce my ADRD risk by quitting smoking based on my age, blood pressure, and health history? A second patient is interested in, I have some cognitive changes. I want to know when to see a doctor. And a third one tries to find matched clinical trials to join. Each care task is supported by a specialized AI system with task-specific tools, models, and data. In the early intervention example, our agents go beyond fixed knowledge and generic literature summary. It's grounded by personalized digital twins, real-time data analysis, and traceable reasoning. On performance, our risk prediction model has an AOC over 0.9, significantly outperforming GB5 in benchmarking. On feasibility, our models and digital twins are developed by longitudinal data in ADRD domain. For example, over 12,000 subjects, ADRD patients in the UK Biobank. These models are made available through our platform on AD Workbench, build a direct path from data to care. On the researcher-facing side, we build a pipeline to link with AD data foundations, along with domain-specific tools and models. These resources support specialized AI agents, cover major data types in ADRD, such as EHR and neuroimaging data. Everything comes together in an agentic AI framework to support our AI co-scientists work on a wide range of research questions, such as biological agent in ADRD research prediction, multi-organ connections with the human brain, and treatment options in personalized therapy and drug repurposing. Our AI co-scientists can help human researchers to work on this question much more efficiently. For example, in biological agent, our AI co-scientists can coordinate the full research workflow, study design, data analysis, reporting, with a human researcher in the loop. We have aggregated biological agent clocks across over 10 human organs and multiple data types, so any researcher can directly access multi-organ insight into biological age and their implications in ADRD risk, without knowing deep expertise in every data type and every organ context. Our principle is simple. Each agent is built by domain experts with the best possible data, and each researcher can use these tools directly on AD Workbench without starting from scratch. Now, I would like to highlight one of the key innovations of our platform, the cross-layer integrations. It works at two levels. At the agent level, our patient care AI agents and the ADRD research co-scientists, they are built to work together in the backend. Our platform enables agent-to-agent communications so the researchers can exchange ideas, data, information, and work on the tasks from both sides. At the user level, our platform transforms physician-scientist collaboration. Here's one concrete example. A physician is seeing a patient with a complex case, multiple risk factors, other health conditions, and there's no clear path to treatment. Just like ordering a lab test or cognitive test to get more insight, the physician can request a personalized data analysis on our patient for this, on our platform for this patient. And the patient care AI agent will receive this task and coordinate this data analysis using the patient's own health record and the latest research data on the platform, working with AI co-scientists and human researchers on another side of the platform. And together, they will deliver a personalized evidence report for this physician to make a clinical decision. We will show a few examples in the live demo, but there are many more opportunities for physicians and researchers interact through our platform, which is made possible by this type of new AI approach. The impact of our product is clear: personalized, accessible ADRD care powered by real-time science. Most current AI tools treat patient care and scientific discovery as separate problems with little exchange between the two. We aim to build the first AI platform, integrate care and research, and closing the long-standing gap between clinical practice and scientific discovery for ADRD. Our roadmap is already on the way. Within the next 3 months, we will finish the deployment on AD Workbench and launch open beta testing. Oh. Sorry, I haven't done that. I can just talk maybe. And after the testing period, we will release the first formal version of our platform on AD Workbench. And in the long term, it would be a great opportunity to work with the AD Data Initiative to deploy our AI products across the hospitals and research institutions across the established platform. This will transform care for millions of ADRD patients and spark many new ideas how physicians and researchers can work together with AI. Now let's welcome Eddie to do a demo of our product. Thank you very much.
Hi. In this demo, we will present OutSage by Cortex, an agentic AI ecosystem that connects ADRD personalized care with scientific research. Here, in our portal, we can start either as a patient or physician, or as a scientist. First, we will show how the patient and physician portal works. A user may start by uploading their health profile and then access different personalized care tools shown on the left, powered by our AI agents. First, the risk assessment tool predicts the ADRD risk progression across a specified time horizon. For example, from cognitively normal to mild cognitive impairment within the next 5 years. The output includes this numerical risk estimate, a short explanation of how it's generated, as well as the selected supporting literatures. Now, after patients and physicians know their risk, they can have a more systematic understanding of their risk profile with our digital twin tool here. We can build a digital twin, and the report includes key health metrics like multi-organ aging and health conditions, as well as risks for ADRD and related comorbidities. Now, having a good understanding of our risk, we can start exploring potential intervention strategies using our early intervention tool. Here, if we're interested in how moderate exercise may modulate our ADRD risk, we can simply select this option and update our risk score here. Now, on the right, the reasoning steps are shown in detail, showcasing key interactions with our AI agent. Importantly, this analysis is conducted in real time using latest research data. Great, now the analysis is done. We may review the results that includes this updated risk score from the intervention, as well as the corresponding analysis. This includes analysis details, key findings, and the detailed analysis code for your reference. Importantly, this analysis is also tailored to the user using only the user's personalized digital twin. And this tool will be especially helpful for physicians to recommend early intervention strategies to their patients. Then we will take a look at the cognitive assessment tool that patients can use to understand their cognitive health. And this will also help with early diagnosis of AD. If not diagnosed with AD before, the patients can complete some basic screening tests. And then our AI agents will determine whether a formal clinical visit is recommended. If yes, available care providers will be recommended below. For ADRD patients, on the other hand, we can explore clinical trial opportunities with our trial opportunities tool here. Our AI agents will find matched clinical trials based on profile similarity, and patients may then find similar clinical trials to participate. Finally, the care management helps us evaluate and predict ADRD-relevant comorbidities, therefore supporting long-term care management. Now, we will move on to our first interaction scenario that connects patient and physicians with scientists. In this patient and physician portal, we have a care assistant window that allows physicians to ask health-related questions. For example, if a physician is interested in whether a patient with problems in multiple organs having elevated risk of ADRD, our care assistant will first provide this preliminary answer. If this is not enough, we can forward this question to scientists for in-depth analysis. Now, we can switch to the scientist portal. Here, in the research inquiries section, we will find the newly submitted question from the physician, and scientists may start conducting research relevant to this question. Once started, a new AI workspace will be initiated where scientists can interact with our AI agents to conduct the analysis. Now, our AI scientist is processing the question, understanding it, and then will generate a detailed analysis plan for scientists to review. Great. Once the plan is reviewed and approved by the scientist, we can just say yes. And then our AI agents will just conduct the analysis automatically. On the right, all the intermediate outputs and logs are shown. Notice that this analysis is also conducted in real time using latest research data. And also, this is a coordinated effort between many specialized AI agents that is done through our AI co-scientist. Finally, once the analysis finding is done, we can review this compiled analysis report here, and scientists may send the report back to the physician. Now, if we go back to the physician's portal, we can find in the scientist Q&A section that the updated findings from the scientist are shown here. Well, now we can review the findings as a physician and provide more feedbacks with the scientist here. This workflow will be especially helpful for physicians to evaluate complex patient-oriented conditions and make important clinical decisions. Next, we will move on to the second interaction scenario, demonstrating how scientists can interact with physicians to initiate their own studies. So here, our AI-powered research planner can help scientists initiate and manage their own studies. It's very simple. Just enter some study requirements. Very simple study requirements. And then first, AD Data Initiative will recommend some relevant data sets. If that's not enough, we can generate this detailed study plan and publish to collect more data from patients. And now, besides this website, our platform is also fully integrated with AD Workbench environment. Here, any user can just start a Jupyter Notebook and access the same model point we just demonstrated. The workflow includes just some setup, input data, loading and calling the model endpoint, and then the output will be generated. Here is just an example of the risk assessment report.
Thank you. Well, thank you for the presentation, really compelling, and I'm going to open this up for questions. Yeah.
May we switch to the demo page?
Hello, can you screen to the QR code?
Yeah, demo page.
Thanks for the presentation. Great. One of the goals of this work is scalability and ensuring that we're accessing globally, providing this solution globally. What barriers do you envision, especially as we think about patient care and accessibility, even for the physician, into this ecosystem? And how do you envision scaling this globally to maximize the input of this two-sided, dynamic of the patient, the scientist, and the physician to support the patient?
Thank you very much for this very insightful question. And we think one of the key things we need to consider is we have to, the product have to be under regulation, which means maybe the best possible ways to integrate with, for example, establish a health network, for example, the hospital networks. So there's already under regulation, and we can do supervised deployment. That's why we think it will be a good opportunity to work with AD Data Initiative with the established network. Thank you.
Thank you, really interesting. Can I ask, so this link between, again, from discovery to the clinic, have you had data that's linked to be able to train and validate these models on linked medical records and kind of, more discovery data, more research-based data?
This is a great question.
Can you tell us a little bit more about that?
Yes, thank you so much. This is a very good question. So when we train the model, we collect data from both two contexts. First is the large-scale biobank, which is mostly for research. And the second is the clinical setting data. And one example from the biobank is the UK biobank and the pre-medical biobank. And the second one is the mimic data from Boston. And they have clinical setting data help us to train the models.
Okay, and if I could follow up, what about diversity? Because of course, if this is going to go to the clinic, you need, UK biobank. Right?
Yes.
Does that have a diversity, or do you have other data? Or
yes, this is a great question. And honestly, mostly now we focus on data we already have, including the UK biobank data and data from Philadelphia, pre-medical biobank, and some data from Boston, and some open data resources around the US, like the HCP agent. And in the later, we try to have more data and increase our generalizability. Thank you.
Just a follow-up question on the data and how you go from UK Biobank, very diverse and significant, to micro experiences. So if, as a physician, I want to upload the data of the patient, how can I make sure I do that through APIs, but also what's the level of capability I need to have from a technical perspective to use the tool?
Yeah, thank you so much. That's why we thought maybe the most feasible ways this tool can be integrated with, for example, the Epic or Oracle, the established electronic health record system, so the data connection and the transmission will be very smooth. Thank you.
If I would ask you what data sets that you would like to have that you don't have now, what would they be?
Yeah, I mean, we want the more data, the better, like global data.
Specific data sets that you thought of?
Yeah, we are thought about some data. We see some research paper like from Latino American, and so if we can get the data from that network, that would be great to increase the generalizability. And honestly, we are open to, for example, people data from Asia and other big hospitals can contribute that data.
Follow-up question, do you think this is a regulated medical device?
It's a very good question, and I mean, as a whole, definitely, I think it should be regulated. But some little functions maybe not need that strong regulation, because some are just like recommendation of like lifestyle. For that part, it might be fine. But anything involving in clinical decision, we think it should be seriously regulated.
Thank you so much for your presentation today.
Thank you very much.
Thank you.
Okay, we have the pleasure of welcoming Biomni AD to the stage. You have 15 minutes for presentation, followed by 5 minutes of questions.
Hi, everyone. I'm Kwan, and an associate professor of genomics and AI at Mount Sinai. I'm here with Dr. Kesing Huang, Stanford Computer Science PhD, and we're proud to present Team Biomni AD. Our team is composed of a list of established computer scientists, Dr. Yuri Lesovich, professor of computer science, whose lab has developed some of the most widely used tools and approaches. My colleagues, Dr. Tawfiq Raj and Dr. Bin Zhang, whose lab has generated some of the highest impact, most cited data in Alzheimer's disease. And Dr. Henry Miller and myself, both practitioners of applying AI and machine learning approach to biomedical research, as well as founders for non-for-profit organizations. It is our privilege to be in this room with all of you experts that are dedicated to resolving Alzheimer's disease. And we're really excited because we bring to you BioMni-AD, your AI co-scientist that can help turn your Alzheimer's data into your next big discovery. We're a fully open source project, already have thousands of GitHub stars and users and an active open source community, so we allow you to do that in an open, transparent, and validated manner. Alzheimer's disease is a really troubling villain for all of us. We haven't solved the disease because the disease has turned out to be much more complex than we thought. But we might already have the answer in the data. Over 30 years ago, my predecessor geneticists have found 3 genes that point really strongly to the amyloid beta pathway, for which we have some evidence for disease. But it turned out that for many of us, and even in the Mendelian disease individuals, the disease is an interaction of hundreds of genes, many different brain cell types, and even peripheral cell types from other tissues, as well as other environmental factors that come into play. And so this disease is much harder to solve than we think. But we also have the best data there ever is. We have genetic sequences of over 1 million people, tens of millions of full expression from brain cells and outer cells relevant to the disease, tens of thousands of people with their proteomic blood biomarker data, with clinical data, imaging data, etc. However, to bring all together all the power of these data sets, each of the data sets have different kind of sampling intricacies and difficulties, and also not to mention that they're across different modality of data takes years and months, not to mention to train an expert, even distilling insight from 1 million people's genetic data into insight could take 5 years to train a PhD, and their time is extremely limited. So we think every Alzheimer's researcher is already a brilliant detective, but what they really need is someone that can work with them to complete a full view of the disease, aside from the evidence that they're looking clearly at a time. And with that, we hope BioMni-AD is one of the solutions. What is BioMni-AD? BioMni-AD is born from BioMni, which was an open source project that was published in the summer last year from Stanford University. As I mentioned, it already has a lot of open source community, over 2.7 thousand GitHub stars, thousands of people on Slack actively contributing, asking questions for feedback. With that, we built a specialized fork, BioMni-AD, to work on Alzheimer's research. It is very easy to use. All you need to provide is a simple natural language research question. So for example, if I want to find a new microglia targets, based on the mapping of the genetics from the large resilience studies, where we find a lot of people with, say, high-risk genotype like ApoE4 that develop the disease, that versus those that end up being really resilient, it will then find that GWAS, find that gene mapping data, and then conduct the analysis. BioMni-AD is able to retrieve from the database of over 180 tools and 60 databases. And these are over 30 more than what we presented a few months ago, making it a fully capable Alzheimer's disease research assistant. BioMni-AD is also capable of reinforcement learning if you have expert traces that you think particularly apply to your specific domain of problem. Since just a few months ago, BioMni-AD also improved performance based on parallel tool cooling, better data caching, and then a new interface which allow the user to review the plan, to either revise or approve the plan before the agent proceed, which the agent will then proceed to generate code, run the code, look at the observation, troubleshoot like a human analyst would, and eventually produce output along with all the analysis code and traces along the way. Now we have the hero. Where do we go from here? We think that we really need a really good map to work alongside your co-scientists and yourself to track down Alzheimer's disease. And we think the best data is really the deep patient data, many of which we have generated at Mount Sinai. Since we last presented a few months ago, we have doubled the size of our expert-curated Alzheimer's database. And these are all agent ready to use. Agents know exactly what a data set is about, what a data format is, so we can effectively generate code to analyze that data. This, we're very excited to say that it also includes already 2480 workbench data sets that we have obtained access to and make sure that the agent know how to use these data sets well. And this covers a wide domain of data, including genetics, biomarker, QTL, CRISPR, single cell, different brain regions, different sex data. Among which, in the 80 data lake, we also particularly infuse in our Mount Sinai generated data sets. And so just give a few examples. RADAR, the largest rare variant collections, validated in over 1 million people's whole genome sequences generated from my lab. Mount Sinai Brain Bank, which we have the brain, different regions, multi-omic data generated in Dr. Binzang's lab. MIGA, largest collection of microglia-specific cells, omics, single-cell omics, generated from Dr. Tawfiq Raj's lab. So with that, we believe the user can then smoothly go with BioMni-AD to all this big data, generate different kind of research milestones they're interested in, and hopefully go about at Alzheimer's at different angles, coming up with brilliant solutions. How well does it work? Our team really cares about this question because now there are a lot of AIs and agents that are able to help you, and really want to make sure BioMni-AD is worth your while. We go out of our way to show that BioMni-AD can reach expert-level performance, including that we already showed last time it's able to reason well over database biological sequence, cloning, plasmid design in the state-of-the-art manner comparable to human experts. In addition, and importantly, for real-world research tasks that us computational biologists or bioinformaticians will do in lab, including GWAS fine mapping, CRISPR, or single-cell analysis, it do much better, actually 400% better than your baseline LLMs like ChatGPT or like your Cloud Sonnet models, because it knows how to use these data sets and tools like an expert would. We also specifically built AD machine learning benchmark called BioML Bench, and we found our agent to be the first place agent among all across all the open source agents. Lastly, to demonstrate reproducibility, we curated the literature really carefully to find a set of retracted disease targets and a set of comparable non-retracted targets, and we blinded BioMni-AD from any literature to conduct CRISPR data-only analysis. With that, we're able to show that BioMni-AD's finding, just based on the CRISPR's analysis, is 17-fold more enriched than the retracted disease targets, showing that it's really able to generate robust scientific insight and even find disease target that you can really trust. Where do we go from here? BioMni-AD is fortunate to be born from an open source community that is already really widely active, and we really want to give back and enhance this community. BioMni community, as I mentioned, already has over 12,000 users across 4,000 organizations, partner with Amazon, Anthropic, SC First, and used by all the top pharma, biotech, and academic organizations. From here, we're really interested in deploying on AD Workbench, which already has happened, but expanding it to be able to use all the over 300 data sets that's up there and provide some free usage for users to get used to the platform. We also are ahead of schedule and actually already launched the adaconsortium.com, which you can visit, where users can submit their agent-driven discovery. So if you and your agent partner together and have a discovery, there's going to be so many of this coming up in the future and probably already, and we want the community to come together and review and upvote these so that we will identify among all these agent-assisted analysis which one are actually the most meaningful, among which I hope with our prize money we will be able to validate in our established iPSC-derived neurons and microglia already established at Mount Sinai. So these include targets or compounds that we can help validate and really help translate these not just from in silico agentic findings, but to actual biomedical breakthroughs that can help. Our goal from here is having 80% of Alzheimer's researchers using BioMni-AD, and we believe that can save hours every day and turns data into discovery much better. So with that, I'd like to show you Biomni AD in action. And so here, you can see our Biomni AD agent working on the AD workbench. It first scans all the data sets on the AD workbench and our data lakes. So you can use Biomni AD just like how you use GPT, very simple. I'm going to ask, recently it was found that the shingles vaccine is very preventative against Alzheimer's disease, actually reduced risk by 20%. I'm really surprised by that findings. So I'm interested to find out shingles vaccine affected pathway. How does that intersect with AD signatures? And then with that, you can see the agents will go into scanning specific context of this question, retrieving the relevant resource before it starts to carefully formulate a plan before execute. And really pay attention to this step because I think the plan is the most important part. The agent can go on and execute, but you don't want to go on the wrong route. So here you can see that in the data set that's integrated and scanned by the agents, these are the AD workbench data sets. We also have our Biomni AD data lake that is infused in from Mount Sinai and other data sources and other public data sets. Now let's go and see when the agent runs some of the previous analysis of Biomni AD agents. You can see that all the output files are saved into specific run that's dated and foldered with their code. And the agent provides a final analysis. You can see that the agent has taken many steps, analysis along the way, generating some figures, using some code. You are able to troubleshoot as the agent runs along the way. And every step, the agents think about what they find, observe, and then generate the next code for the next run. Let's go back to the first agent, and you can see that it already come up with the analysis plan. It's going to scan the local data sets to identify and create the AD gene signatures, curate the signatures from the vaccine, perform analysis, and that's just approved from here for the interest of time. When we do that, we can go look at another example where it also conducted analysis and look at the code, et cetera. And this is another example here across different kinds of analysis. This was building a proteomic model to predict Alzheimer's disease 10 years out, and you can see that it has all kinds of metrics, et cetera, that's being generated. The great thing for the user is that you are able to then go to the folder of the run and actually look at not just the figures, but also specifically the Python notebook that contains all the code as well as summary statistics generated along the way, so expert can really validate and modify if needed. Let's go back to look at the first agent. It's already running multiple steps. I'm not sure within the interest of time it's going to be able to finish, but to close off, let's look at the proteomic model that the agent completed plan. It was exploring these data sets from 545 patients, parse these files, able to do these analysis, have the metrics available, and eventually have all these output actually completed in a specific folder. With this analysis folder, you can then go see that we have already have many runs of these. That one was just right here, and our current running run will be right here. So with that, that's our Biomni AD agent hard at work, and we're really excited to see what you are going to be able to do with the Biomni AD agent. Thank you.
Thanks for the presentation. Can you speak to how the interface between the BioOmni platform with the BioOmni AD agent and project, and that as there are improvements to the overall platform, how does it influence what you develop for Alzheimer's disease?
Yeah, so the original BioOmni project as released last summer has a Gradio user interface, and I think that interface was a little more technical. Since then, we overlifted to this chain-lift interface. And we believe that given users are so comfortable using GPT, cloud, online all the time, this is one of the best solutions given that we have all the files tracked anyway, so the user can go to the folder if they are more technical to visit that. And so in our experience, and also have worked with many users, a lot of users actually prefer this kind of interface. They can click open the code if they want. It's not up now, but they can click open the code if they want to supervise along the way, but they can also more interactively chat with the agents to plan and approve plan and then execute the analysis.
Yeah, and also to add on that, so all the improvement based on the open source Biomni platform can also be easily ported to the Biomni AD. But the key benefit, I think, from Biomni AD is all the data sets from the ADDI and from all Mount Sinai, which really contextualize the agent. So we think it's a perfect combination. Biomni has the best agent out there. They have the best data sets, and then you integrate together, you can do all kinds of different analysis. Yeah.
Yeah, question from one of our judges observing remotely here from Tom. Recognizing that the Biomni platform, you have an open source and you have a commercial as well, and you've integrated this in the ADDI platform. Can you tell us a little bit about the plan for your ongoing improvements and maintenance of this particular open source going forward, should you win the prize?
Yeah. So right now we're competing with this open source version. Kazin has founded a company that has Biomni Labs, which is a different product. And this open source version, the Biomni AD, is completely open source, will continue to be open source. I'm planning to integrate more of those data sets. And as I mentioned, I think the development, given a really active open source community, one of the key strengths is that when a user notices one of the tools they really want to use is missing, sometimes they will tell us through the Slack channel, and sometimes they will build it themselves. And so that part is very organic, and it really just happens. The part about deployment, user tractions, and I think now we're going to the next phase of community curation, is what I think will really be critical to move really into translation. And that's why I emphasized in the end a lot about the price allocation into not just the AD Workbench deployment, but also the consortium. We already actually awarded cash prizes to the first entries, but we really want to bring and incentivize the top entries coming in and collaborate with Mount Sinai to really validate the top genes, the top compounds, so we can really move towards translation.
So what are the safeguards, the embedded safeguards for when an agent either fails or when contradicts prior knowledge? Of course, the human in the loop, but what other integrated safeguards have you put?
Mm-hmm. Yeah, that's a great question. The agent has reflection built in, so actually, if we pop up the terminal, sometimes the analysis will fail, like any human writing code will fail, and the agent will reflect on it, observe it, and be able to normally recover. It's really the extent of a problem, a coding problem that an agent can complete, then it will fail to run and you can go to the folder. It really rarely happens. Normally, I think, frankly, the agent tried to do something else that is related that may not be in the direct plan that laid out because that plan, that step that really just stuck. And then that you will be able to see as fully transparent in the output.
Yeah. And also, so we keep it simple on the interface because we want to make the scientists use it easily. But actually, in the backend of agent, we actually added a lot of guardrails. For example, like each step of the agent, we have like another agent looking at the traces and try to see if there's some hallucination, factual mistakes, and it's able to very easily to detect them and self-correct them as well. So,
and just one follow-up question, and you have also biases review and checks for how the plan is built by the agent, yes or no, and who supervises that?
Yeah, and that's a great question, and that's why in this version we really build in when the agent come up with the first plan to let you revise. And just now in the workflow, I didn't say added, but I could add the plan directly because I think it's very important for the agent to really pursue what the user believes is the right question.
Follow up on Does the system refine hypotheses by itself? Does it do any experimentation? How does that work?
Yeah, so it depends really on the user query. We really give a lot of power to the user. So, just now, actually, my question was a little open-ended. They could have formed very different plans from what I perceive. I can also give a very specific plan, then they will be more similar to what I already planned out. And again, I think that's why the approve or added plan phase for us was very important in this iteration, because we really hope that the users can come in really broad or they can come in very specific, and we hope that the agent will be able to really deliver what the user wanted.
Thank you very much for a compelling presentation. Let's thank Team Biomni MD.
Thank you.
All right, welcoming to the stage for our final team is Prima Mente. The stage is yours. 15 minutes of presentations, 5 questions.
Amazing. Thank you very much, everyone. I was going to say, with the last presentation, I thought I'd maybe rush through, but the 8-hour time difference means I'm actually really awake. So I think we're going to keep the full 20 minutes. So Jonathan and I are here from Prima Mente. We're going to present to you something a bit different, actually. So Parthenon is our solution for the future of ADRD research for the community at large, and we're excited to show you our progress since CTAD and our vision for the future of research in this world as well. So the primary bottleneck I think we can all agree on in this space is our inability to rapidly validate hypotheses and potential pathogenic drivers for disease. Today 2 things are happening in the world. First, almost everyone is generating large biological molecular data sets representing health and disease. In fact, the vast majority of data on AD Workbench is molecular in nature. And the second is that there's an explosion in the number of scientific hypotheses we are deriving from AI models, whether they may be frontier models like from OpenAI, Anthropic, or the specialist models that have been presented this morning and this afternoon. But there is the status quo is still troubled by 2 things. Animal models don't translate to human biology. We've treated a mouse 1,000, 10,000, 100,000 times over probably with AD. And second, while cellular models show great encouragement and possibility to model disease in vitro, they are very complex, costly to scale, and almost artisanal in nature, limiting access to researchers. What we need to, say, escalate the degree of research in the space is to provide every researcher with the ability to run near infinite experiments at scale with limited cost. This is where Parthenon comes in. Parthenon is our integrated modeling and discovery platform for in silico research. At the backbone of Parthenon is world-class ML and data infrastructure that brings advances in architectures, foundation modeling, and data processing from our team to every researcher in the community. There are 3 core components. The first is our virtual cell model hub, which allows everyone to generate bespoke biological virtual cells from molecular data sets. These domain-specific models are really critical and are required to go beyond the reasoning models that are available today. In addition, this allows every researcher to either use pre-existing AD Workbench data or bring their own data to the table to derive and build bespoke virtual cells that can either act in isolation or in networks with other virtual cells to simulate experimental environments. Our training studio allows researchers to utilize the best-in-class methods that we know from foundation modeling, including fine-tuning, post-training, and reinforcement learning of the models. And finally, our discovery engine, which allows every researcher to utilize these tools together for data exploration, hypothesis generation, and inference of the models as well. Since CTAD, we've generated a new co-scientist named Athena, after, of course, Parthenon was Athena's temple, so we named it Athena. So Athena is the co-scientist that drives a lot of this. And the other thing is our fully autonomous wet lab, which we've embarked upon since CTAD as well. Every experimental discovery with AI needs real-world wet lab validation, and we will be able to develop our lab and utilize it as a tool for every researcher in the world to connect it from Parthenon into a real-world wet lab experiment done autonomously in London. Our lab design has been complete actually this week. Construction will start imminently and be live at the end of Q3, and we'll make our lab available to the community through Parthenon and the AD Workbench. This will unlock the better together story here, which is this flywheel for recursive self-improvement and the race to the top for discovery, where data will drive the derivation of new best-in-class biological models, creating new biology, creating new experiments with real-world validation in either our lab or the world's labs to drive new data outcomes. Our goal is to democratize ADRD research for the community, including modeling and lab-based validation of experiments. These numbers are striking, but they are true, that the acceleration and drop-in cost of discovery is it's thrilling, right? A 50,000 times reduction in cost to screen a target for a perturbation, and a 40,000 times reduction in time to screen a target as well. In addition, we allow the capabilities for everyone to also build new models as well from scratch, comparatively normally 30 hours if you have the capabilities, to minutes themselves.
So data and modeling kickstart this flywheel. Data provides the information, and the modeling allows us to unlock this for biological discovery. Since CTAD, we have made rapid advancements on both of these fronts. On the data side, we have orchestrated a global data consortium by expanding existing data partnerships with academic labs. This has allowed us to ground our models in real human biology. Additionally, with these partners, we will add data to the AD workbench so that we can solidify the AD workbench as the data backbone for the community. Additionally, we have aggressively pursued de novo data generation. We are embarking on a 1 billion cell perturbation atlas for ADDI that will include chemical perturbation, genetic perturbations that also include double perturbations, and critically cellular perturbations, where we can study cell-to-cell interactions using our partnership with Celinome. This data set will allow us to expand mechanistic discovery and target discovery, but critically it will also allow us to build towards a network of agentic virtual cells so that we can solve this disease as a network problem rather than a single cell type problem. Data is just the first unlock though. We differentiate ourselves with how we build our models. Our models are built on molecular data, and we have unlocked generative modeling since CTAD. Together, this allows us to do de novo predictions on synthetic cells or unseen cells. We can do cell annotation with label transfer, and we can do combinatorial perturbations as well. With what we have, with the way we build our data, we're at the frontier piece of what we can do with virtual cells. And because we build our models on molecular data, we can do what LLMs can't do. And the results are incredibly promising. Since CTAD, we have pushed forward with our microglial results, but we have also showcased our ability to expand to other virtual cells. We have built an astrocyte virtual cell as well. For a refresher on the types of modeling that we have forward and inverse modeling. What this means is forward modeling, we ask the question, given a cell in a specific state and you add a perturbation, what's the outcome state that will occur? Inverse modeling asks, if you want to shift a cell from one state to another, in particular from a disease to a non-disease state, what perturbation do you need to add to cause that shift? On the forward modeling side, we have shown that our models are grounded in biology. On the left, you see observed log2 fold changes against predicted log2 fold changes for differentially expressed genes, and we have a Pearson correlation of 0.96. It's incredibly high concordance between our models and observed. Our models understand biology. On the inverse modeling side, we've shown a few things. The first is that we have improved our models since CTAD. With the microglia in dark blue is what we are currently doing, and in light blue is what we had at CTAD. We have improved results. Very importantly, though, the results themselves are already predictively powerful. So for the top one, essentially what we're doing here is asking the model to say, if we want to shift from one cell type to another, which of the ranked perturbations is the one most likely to do it? And we have a 59% accuracy currently. To put this in perspective, if we were going to do this in the wet lab, we'd be excited to get even 1/10 of that power. So our models are currently capable of doing amazing predictions, and as we get more data and expand our modeling capabilities, it will get even better. On the astrocyte side, there's a couple of things that are important here. One, we have unlocked astrocyte virtual cells. Two, we have shown the ability to fine-tune on existing AD-RD data sets, the type of data that exists within the AD workbench, to improve our models. So we can immediately action on those types of data to improve the models that we have. What does this mean? It's now time for us to put Parthenon in the hands of the global research community. Within the next 3 months, we will release Parthenon to AD-RD community through the ADDI, through ADDI. We will expand our models to include multiple neuronal subtypes, and we will enable joint cellular modeling in one in silico environment, creating these virtual cells and interaction of virtual cells to scale towards whole cultures, organoids, and ultimately synthetic brains. Using our wet lab, we will experimentally test 50 of the community's discoveries using Parthenon and provide this back to the community.
So the idea here is that we have every researcher basically having a digital wet lab on their own laptop with the infrastructure to do modeling and scaling of discoveries themselves. And there's a flywheel here where as every lab generates new data, the models improve, the models get better, more data is generated, and it truly is a race to the top. Bose this morning outlined his vision for AD Workbench as a collaboratory, and we think by combining infrastructure on data generation, results generation with data infrastructure, we can genuinely create this global collaboratory environment.
So now we will showcase our demo and what we think is possible when we integrate Parthenon into the AD Workbench. Few important things to note beforehand is that our perturbation modeling is live. It's done with real data and with real models. Second is that we've deployed an app using Azure VMs that have been provided by the ADDI folks so that we can show that we can technically integrate. Parthenon is our model building and discovery engine that we've already shown is capable of biological discovery, but we want to show how it's also able to unlock each researcher to action on data that already exists within the AD Workbench and feed that back into the community. One of the key pieces is our Model Hub. Model Hub allows you to view any published models, publish your models yourselves, compare them, but not only is it an exploratory tool, it is also a tool for inference. You can go through and connect to our discovery engine. We will show how we do inference with our inverse model. There are two different ways that you can select target and source cells. The first has been seen before with UMaps. Come back to this in a moment. We've added a new feature called Label Transfer or Reference Atlas, where we have transferred labels from an existing data set that has been curated by the community. And you can select your target and source cells this way as well. For the purpose of the demo, we'll go with the UMap. We will select cells high in a disease-associated score as our source, and then we'll select cells high in an antigen-presenting score as our target, and run inference. Our model shows that ARID2 in this case is the most likely perturbation to shift between those two populations. This has been seen before. What we've added now is a co-scientist that we're calling Athena that allows us to interpret the results. There are 3 steps to this. There's analysis, there is augmentation, and there is automation. One thing I do want to note real quick is that you can select any model to power this. We are currently using the DeepMind AI co-scientists because we are developing a partnership with DeepMind. But the first step, analysis, essentially does a literature search and gene signature analysis to identify genes upstream and downstream of our target. The augmentation tool then uses the pathways and genes found to identify CRISPR genetic perturbations that could be used and used for lab experimentation. But if you're not interested in CRISPR and you want to do drugs, it can also search existing drug databases to identify potential candidates that can target the pathway that was identified previously. From here, we can go to automation. During automation, our co-scientist searches the AD Workbench to identify data sets that might have the perturbations of interest identified previously. And we're showing an example of what that would look like here. You can take one of those data sets and immediately fine-tune on it and going into our training studio. This provides access to ML expertise provided and powered by Prima Mente to those who might not have traditional experience with ML training. And once this is done, you can run evaluation and publish to the Model Hub. When we publish, this is when each person's discovery becomes the community's discovery. So there's actually another feature that I wanted to touch on here. Because of the new architectures that we mentioned before, we are able to do generative modeling. We can visualize what's called a vector flow field from our models. Essentially what this is telling us is the effect of a predicted perturbation on any source cell. This is not just cells that we've seen in our data set, it is any cell. This can be a synthetic cell. On top of this, though, we can also do combinatorial perturbations. So this asks, if we don't have a single perturbation that can reach our target destination of interest, is there any combination of them that we can search through that would yield this? We're running live inference on this at the moment. It's checking all possible combinations. When this is done, it will give a ranked list of the combined perturbations that we could use. And what this does for us is it massively expands the search space, and it allows for experimentation that would not have been possible without our in silico lab. Because we connect this to the Model Hub, this means that each of our learnings becomes the community's learnings for Alzheimer's disease. Yeah.
Thank you for a compelling presentation. It's open for questions. Perhaps I'll start with the first. Can you describe, really fully featured tool here. Can you describe who your target first users would be, ideally, of this?
You want to go?
You can go. I'll follow up.
So I think the first users are likely going to be experimental scientists who are trying to identify potential targets for several things, either therapeutically targets that could shift cells from like a disease state to non-disease state, but alternatively it can be used also to shift experimental models to more accurately represent human biology. And so if you have, do the label transfer that we mentioned there, you can start to identify perturbations that would shift those experimental models to more accurately represent the distribution that you see in real human biology.
Yeah, in parallel, we also feel because you'll be able to have multiple different cells from multiple different protocols and environments and reconstructions of human cells as well from human tissue profiling, anyone that does a wet lab experiment could utilize this alongside to validate other protocols of cells to get concordance of the results against different experimental models.
Thank you. You talked about virtual cells, synthetic data. Have you kind of dealt with biases, or how do you deal with biases in this data? What is the proportion of real to synthetic? How do you kind of juggle this?
So all of the data that we have trained on is real experimental data, if that's what you're asking. The synthetic component that we are discussing is because of the generative nature of our models, we can essentially predict the impact of a perturbation on a cell that falls in any distribution. And so when we were clicking on that flow field, that's essentially saying in embedded space, if we had a cell right here, what is the effect of the perturbation on that, even if we haven't observed it so far.
Critically, this is also why our lab in the loop, or an agentic lab in the loop, is really important for validation of synthetic discoveries. But yeah.
Have you been able to do any validation?
Yeah, so I was hoping to have it today, actually. We had 3 targets we discovered. The experimental work we've done in our lab, actually, we've found some results. I can show you them afterwards, actually. There's some alignment as well. We're able to predict the perturbations are pretty good, but we want to do far more at scale.
Thank you.
As the field is rapidly evolving, how will you ensure that you can orchestrate and optimize across all the models that are out there as that goes by the day, by the week, by the month? And it's really critical for the results that you want to obtain.
Absolutely. So behind the scenes, there's a lot of AI modeling infrastructure that we build internally, whether that's novel architectures or ways of evaluating performance of models. On the back end of all of this will be versioning of different architectures behind community-created virtual cells or virtual cellular environments and performance against evals as we improve them internally. So then we imagine, what you'd see in the normal model space, versioning of different models that then get retired over time based on performance as well.
Given in the field of iPSCs, you can really create all kinds of cell models based on individuals with different genetic backgrounds. How do you think about it, given there's so much you can do in the modeling, but there's also so much that you can do experimentally? How are you thinking about bridging different models, iPSCs, based on human-derived patients, and then leveraging the perturbations and the modeling that you have on the platform?
So in many ways, that's actually the beauty of the platform, because you're not restricted. We envision a world where researchers publish their iPSC-derived, say, microglial atlases with any variety of protocols with any genetic backgrounds. So as researchers upload, say, the genome of the cells that they've developed their models against, then it allows us to actually even do comparative genomics or comparative modeling against different humans and species as well. Well, humans, not species, but humans particularly in this space. So I think in many ways that's kind of the power, because it will unify the field in terms of giving every researcher every potential background genetically or experimental protocol to do their own experimental work with.
Quick note on that too, the 1 billion self-perturbation atlas that we're doing is going to be across several different genetic backgrounds as well. So as we're building these data sets, we're going to build in the genetic diversity as well.
How do you plan to engage with pharma, and what's the IP model that you are looking to have?
For the company or for the platform?
For both.
Oh, okay, sure. For the platform, I think, our goal is to democratize research here. So we want to give these models to the community to do discoveries. Internally, we also have our own ambitions, and we do our own program development, for example. Our goal is to work collaboratively with pharma for discovery. We view ourselves as very much the AI model expertise player in this space and want to work together on development downstream with pharma alongside internal asset generation.
Thank you so much to the team.
Thank you.
There you are. What do you think?
Thank you. Nice job.
And I want to thank you all here for sticking out the day to listen to these amazing presentations. Thank you all here in Copenhagen. I want to thank everyone watching worldwide for this as well. I'll remind you all here at the ADPD meeting that the finalists that you just saw will be demoing these, and you can get hands-on demonstrations throughout the week in the ADDI booth. So with that, I'll bring us to a close.